nanostructured materials that they can have very, very special properties. For instance, like the feet of the gecko or the coating of the stealth airplane. The nanostructure influences the properties, and the properties are very different from the bulk. So today, connections from supramolecular chemistry to nanomaterials. So we want to make nanostructured materials ordering on the 1 to 100 nanometers. And we want to use the concepts of supramolecular chemistry. Of course, there's also a lot of other methods to make nanomaterials, either with lithography or using physics, the physics of phase separation. So we'll try to use also supramolecular chemistry. And if we look at supramolecular chemistry, then there are 3 important concepts that we can use. One of them is templating, the other one is self-assembly, and the last one is self-organized growth. And these are the people that invented it. An example of a supramolecular unit that then shows self-organized growth on a surface, and it makes a nanomaterial. So we'll first look at nature. Nature can be a big source of inspiration. And we'll also look at some artificial systems, some self-assembled monolayers, and we'll also discuss nanoparticles in quantum dots. We want to use molecular building blocks to make larger structures, see how they can interact and form. If, for instance, if we look at life, then you can wonder, how could we create artificial life? What do we need to make life in an artificial way? A cell membrane. So a cell is extremely complex. It has all kinds of different quantities, different compartments, but the compartmentalization that is needed for a cell. So you can try to make artificial membranes. That is not so difficult. I think that you know that a micelle usually has only one tail, is an amphiphilic molecule, a soap molecule. Things that make bilipid membranes, they usually have 2 tails. They're usually phospholipids. And these tails, they stick together and they make this little ball. We call it a vesicle or liposome. Basically, it's like an artificial cell wall. These tails stick together and the polar groups are on the outside and on the inside. So inside we have like a little ball of liquid. And of course, in these membranes, you can insert all kinds of functional systems. Using hydrogen bonds to organize molecules is a tool that we can try to apply. Of course, next to these hydrogen bonds between the base pairs, there's also the phosphate groups on the outside and also the pi stacking. So these base pairs also have pi stacking. So we also have van der Waals and dipole-dipole interactions that can be important. Another example from nature is the tobacco mosaic virus. So this bar here is 1000 angstrom. Well, it's really a nanomaterial. So it's a little stick, a little rod that's similar to what we saw on the gecko's feet and the coating of the stealth airplane, making a rod type unit with a supramolecular assembly or with another method that can be very functional. It can have very special functions. So that is something that nature does. Now, can we try to mimic that? Can we try to mimic these kind of aspects? Yes, of course, we can try. So we need to use self-organization, like bilipid membrane. So making a vesicle is not so difficult. I even did it myself in my PhD. I made some vesicles and incorporated C60. So just by sonication or by special probe, you can make it. So here we see an example where they inserted a channel into a bilipid membrane, and this channel, it consists of a cyclic peptide. So here you see the peptide, it organizes through hydrogen bonds, and these hydrogen bonded units, they can insert into a bilipid membrane. So in that way, you can make an artificial channel for ions or protons or something like that. Just like nature does, you can make artificial channels in membranes. So that can be very useful. Here we see the cyclic polypeptide and the diameter of about 1.3 nanometers. It stacks through hydrogen bonds. Well, maybe an antibacterial agent. Of course, Jan van Maarseveen, he works on cyclic peptides and he tries to develop new medicine, new drugs with that. Another example of a hydrogen bonded system, well, you see that there's a structural team that we also saw last time, surface organization of the honeycomb, a complementary hydrogen bonding team. If you block the right sides, then it can induce organization in a specific way. So in this way, they were able to organize 9 components, so 3 of these calixarenes and 6 of these green units. So together, 36 hydrogen bonds, they form this, well, this shape, this sort of triangular shape of a supramolecular assembly. Next to hydrogen bridges, van der Waals interaction, dipole-dipole interactions, you can also use metal-ion ligand interactions. So here we see some examples of organized structures. So you have ions, you have ligands, you put them together and they contain information. They contain, of course, structural information. So they are programmed to form a certain shape. So here we see examples. We can make squares, we can make prisms, we can make triangles, depending on the coordination of the ions and the structure of the ligands, of the organic ligands. So metal-ligand interactions, that's another tool that you can use to make nanostructures by using supramolecular concepts. And one special aspect of that is the so-called metal-organic frameworks. They contain cavities, just like zeolites, but we can change the length of this bridging leg that bridges between the metal sides. So the metal sides are here the corners, and the organic sides are the legs of the cube. So these kind of structures, the metal organic frameworks, they can be, for instance, be used to store gases. And yeah, Mr. Yagi, he's a big name in the metal organic frameworks. He basically came up with them. And there's even some applications. There's one company that uses metal organic framework to store gas, and by using the metal organic framework, you can store more, to release it slower, so it's a safer way to store toxic gases. There's also another application that they call it TruePick. It's something that holds a gas that ripens the fruit. So in that way, metal organic frameworks are applied in products, and they are similar to zeolites, but they're tunable. So you can tune the metal corner unit and you can tune the organic ligand. You can, now in general, they are very hygroscopic. They attract a lot of water sometimes, depending on the metal especially. And it can be difficult to make them. Another inspiration from nature, photosynthetic reaction center, light harvesting. The green plants, they basically are important for almost all the energy on Earth. If you look closely at the light harvesting complex 2, you see that there are like circular arrays of multichromophoric units with a dimension of about 5 nanometer. Nature makes rings of 5 nanometer diameter, and they harvest the light. You can wonder, can we try to mimic that? And here we see a more detailed structure of the light harvesting complex 2. At the top, you see 16 of these bacterial chlorophyll units, and at the bottom there are 8 of these green ones. The wiggled lines are the proteins. There are chromophoric units that are embedded in a protein matrix, and they hold them in the right place. If you look at the 8 chlorophylls on the outside, you can say, okay, this is an octagon. So can we maybe make a supramolecular complex or a nanostructure that contains 8 chromophoric units that organize? That would be a very interesting goal, to use these building blocks to make a supramolecular structure that can actually do something. It can harvest light, it can transport the excited state energy. Inspired on the light harvesting complex 2, this work has been done. They put these components together and they react and they form a supramolecular structure. We can call it a nanostructure if you want. If you look at your chemical background knowledge and you look at these molecules, you should be able to come up with the structure that is formed. And so my question to you is, what will be the product? The question is, what is the product if you put these components together with the information that we just discussed? So please give me an answer. Put on your microphone and say something.
Doesn't it depend on which ratio you put them together?
It can, but if you take a 1-to-1 ratio, what would you expect?
Okay, I don't know, but I have to think about it.
Yeah, that's why I ask questions. So you start to think. Or what kind of interactions do you expect to play a big role? Any suggestions here?
Metal ligand coordination.
Yeah, of course, metal ligand coordination. And what is the metal and what is the important part of the ligand?
Platina is the metal.
Yeah, here we have the platinum metal. Here we have the pyridine. The platinum, it coordinates in a square planar fashion. And basically these two groups, they exchange for the pyridine, and then you make a molecular square. This ligand, together with this platinum salt, one side is blocked by the phosphines. The other groups, they are ions, they can exchange more easily. So in that way, you can make molecular squares. The molecules that are used are this pyroline bis imide, so that's a very strongly absorbing unit. It's also used in car paints. And here we have pyrene. Pyrene is attached as the aromatic group here on the sides. It is a molecular square. It contains pyroline imide legs, and on the side are hanging pyrene groups. It can absorb a lot of light and can also do things with it. Here we see a molecular modeling picture. So we see the pyroline imides, we see the blue pyrenes, 8 hanging on the top and 8 hanging on the bottom. There's 20 chromophores together, 4x4x4 nanometer dimensions. And it also absorbs light and it does something with the light. Pyrene to the pyroline, we have energy transfer and electron transfer. And here we see both structures of the light harvesting complex 2 and these molecular squares. So on the same scale, of course, the light harvesting complex is still a little bit bigger and it only does energy transfer, but it is approaching the size and also the photophysical complexity. Building blocks, put them together and make something larger that can do something. This is actually work of Bashak. And here you also see some of interactions with the light. If we excite these blue units, then they can give energy to the red units. So here you see that the emission of these blue units, it is quite short, much shorter than the blue units by themselves, the pyrene by themselves. And if you see that this line, it sort of goes down, and as that happens, the emission of the red units, it increases. So this complementarity of the decay and the rise time, it tells you that two states are interacting and one gives the energy to the other. As the one population goes down, the other population goes up. So that's a signature for energy transfer in these molecular squares. And that's also what's energy transfer is also happening in the light harvesting complex too. Self-assembled monolayers. And that we will discuss now. There is a movie, I could not find it, but Langmuir of the Langmuir isotherms. He probably was the first one who made a monomolecular layer, and he actually made it on a lake. So there's a movie where he drops a little bit of oil on a lake, and he calculated that it would form a monomolecular layer on this area. You see that the waves, they suddenly stop for a few seconds, and that shows this principle. It is possible to make a layer of one molecule thick on a surface, either on a liquid or on a solid. It can be quite simple. So you have a substrate, you put it in a solution with special molecules, and then they organize. They organize on the surface, and you see that the head group of the molecule is important, and also the tail. Especially this picture, it shows that the interaction between the tails, it can be important energy. And the most important self-assembled monolayers, or the most studied, are alkane thiols on gold. So if you have a gold surface and you put it into a solution with alkane thiols, you get a self-assembled monolayers, which can be very, very organized, very structured, very well ordered. You can try to play with the functionalized surfaces. And here we see an example of that. The molecule, here you see it's a thiol, is an SH group with an alkyl tail. But at the end of the alkyl tail, there's another group, but it can be cleaved off. So you can make a self-assembled monolayer on a gold surface. Then you have a spacing between the molecules because of these large head groups. Now you can hydrolyze off this head group, and then you have a charged group. So then you can change hydrophobic nature of the surface by putting a potential on the surface. So if you attract these negative charges towards the surface by making the surface positive, it becomes hydrophobic. It repels the water. And of course here it sort of attracts the water. So you can change the surface properties of a self-assembled monolayer. So that can be very useful. It can perform a function. So there's loads of possibilities to functionalize surfaces. Thiols on gold, they are the one that really work the best, relatively easy to make. But you can also make such type of layers on other metals, like platinum. It also works for disulfide. So if there's two sulfur atoms attached together in a molecule, it can also bind to the surface. And also thioethers. We also studied thioethers in the past to interact with golds. Another interaction that works very well is pyridine on platinum, but also amines on platinum can work. Acid groups, they can interact very well with semiconductor oxides, with metal oxide, like titanium dioxide, zinc oxide. There's also possibilities to functionalize silicon oxide with trichlorosilanes. Pyrene, it attaches very nicely through pi-pi interactions, van der Waals interactions and induced dipole onto graphite or carbon nanotubes. So you can attach molecules to a carbon nanotube by using pyrene units through the pi-pi stacking. So in this way, you can functionalize a surface with a particular molecule, and it is possible to functionalize different types of surfaces. That is important for what we call dye-sensitized solar cells that uses titanium dioxide. That is something that has been developed by Michael Grätzel. So they call it also the Grätzel cell. It is an alternative to silicon solar cells. But the link is that they use titanium dioxide. So you have to absorb molecules onto titanium dioxide. So that works very well with acid groups, but it can also work with other types of groups. Together with Joost, we also worked on these kind of things. So this is two papers from a PhD student from Taiwan, from Hung Cheng Chen. So he used these molecules to put it on titanium dioxide. So it's actually also a novel interaction. But he was also able to put them on a surface. So you get a yellow layer. But if you now, either with electrochemistry or with a sacrificial electron donor, something that gives off an electron, you can also turn it into the radical anion. So you can do electrochemistry on the layer, and you can reversibly change this color and also the properties. There's also applications for this, for instance, to have self-cleaning glass. I was also once at a nanotechnology conference, and there was one guy from Villiers-en-Boch, and his talk was about self-cleaning toilets. A very thin layer in the toilet that keeps it more clean. The lotus effect. So you may have seen these kind of things. This is the lotus plant, and this is a drop of water that does not stick to it. It's also, there are movies where you see people putting glue on a lotus leaf. So it's a lotus effect. And so the nanostructure of the surface, it induces self-cleaning properties. So on the smooth surface, the dust or the dirt, it stays. On a nanostructured surface, the dirt goes away. And well, you see again that pillar type structures, they are important for these properties. So if you take a very, very good look at the leaves of a lotus plant, then you see that there are these little features, and on top of these little pillars, there are more smaller pillars. So this is a representation of the nanostructure of the lotus leaves, and it tells you that the nanostructure is related to the self-cleaning properties. So that is something that we also would like to reproduce, of course. There's also animals that do the reverse, so they actually collect water on their back. There are certain beetles, desert beetle, so all the water, it goes to one place and there it is collected. So you can also do the reverse. So of course, people have tried to mimic this. So they've tried to make something that consists of pillars. And in this case, they are made of Teflon. And just like the stuff that you have in your pan to fry your eggs, this kind of material is used to make a pillared structure that is hydrophobic, so it repels the water. So if you put a drop of water on the surface, it sort of lays like a little ball on the surface. So in this way, you can influence the surface properties of a material. You can make a super hydrophobic surface coating. So you can wonder, how can we do that? How could we make such pillars on a surface? One option is to use nanoparticles. So if you take a surface and you put nanoparticles on positions, then below these nanoparticles, you can grow something like a carbon nanotube or get a reaction going at that particle. So that brings us to the next aspect, which is nanoparticles. So the trick to make nanoparticles is the following. So you take a solution of metal ions, for instance, gold ions, auric acid, and you put in something that gives off electrons, an electron donor, citrate, but also sodium borohydride is a very good reducing agent, so it gives off electrons easily and you can use it. So if you do that, then these ions in solution, they become metal atoms, zero-valent metal atoms in solution. Now they have the tendency to cluster. These zero-valent metal ions, they will try to stick together, so they form clusters. So if you don't have another trick, then you will just get a very large precipitate. But if in your solution there are ligands present that can catch these clusters, then you can make very well organized nanoparticles. So if you use gold ions, sodium borohydride as a reducing agent, and if you have thiols present, then you can make thiol stabilized gold nanoparticles. With citrate, it's just a question of boiling it up, and at a certain temperature, the citrate gives off the electrons, and the other citrate molecules, they stabilize the particle. But so you need something to stabilize the clustered metal atoms. So in this way, you can make nanoparticles that are quite well defined. So here you see all kinds of small nanoparticles, but if you take a closer look, maybe some of them are a little bit bigger than the others. But one very nice application of gold nanoparticles is in a medical test. So if you have very small gold nanoparticles and they cluster together, they form a larger structure, and that is purple. Bluish or purplish. So actually the pregnancy test that forms these purple stripes, they are nanoparticles that cluster together due to the presence of a hormone. It can be very practical. It can have very nice practical applications. So we also worked on gold nanoparticles a while ago, and we used dendrimers. We use these kind of dendrimers with thioesters to stabilize the particles. Metal nanoparticles are actually very, very old. Faraday, in 1857, he already, without knowing what they were, he made nanoparticles, and they are still in museum, the Royal Institution in London. Under the right conditions, these nanoparticles can be very, very stable. So next to metal nanoparticles, we also have quantum dots. So quantum dots are also small particles, but they are made of semiconductors. Cadmium selenide quantum dots. And here we have a very special effect because the size determines the color of emission. So here you see that small particles, they give off blue emission. Large particles give off red emission under a UV lamp. Here you see an electron microscopy picture of such a cadmium selenide nanoparticle. So in the center of the picture, you see little dots. The diameter is 5 nanometer. And these dots are actually the atoms that are in the particle. So you see that it is like a very, very small crystalline, very well-packed region. And together it forms, well, ball-type structure, but it can have facets. So gold nanoparticles, but also quantum dots, they're not super round, but they can have actually some crystal facets. But the interesting thing is that the luminescence of a quantum dot is size dependent. And you may know that even there is on the market a certain type of TV, a television, that the emitting units are quantum LEDs, light emitting diodes containing quantum dots. And okay, so the size determines the emission color, and that is, you can wonder, okay, how does that work? Here we see the absorption of different sizes of particle. So you see that the absorption just shifts with size, and the emission color also shifts. So how can we understand this size effect of the quantum dots? So the emission color, it depends on the size. So you have small ones that are blue emitting, big ones that are red emitting. The quantum dot, it's basically a quantum effect. So you may remember from your first year of chemistry, a particle in a box, wave functions that are fitting inside this box. Now, this is basically the size of the box, but you can also consider it the size of the particle. Energy differences between the levels, they are related to the size of the box. So a smaller box has larger energy differences, so it has a more blue shifted color. So it is really similar to the particle in the box model. It's the quantization of the energy. So you see that a large energy difference relates to a small particle, and it is correlated to a blue emission or absorption. A larger particle makes the energy differences between the two levels smaller, so it is correlated to a transition in the red, which can also be emission. So in this way, the size determines the properties, and the size is in the nanoscale. At Aldrich, you can even buy these kind of particles that are giving blue to red luminescence. So we want to use the concepts of supramolecular chemistry to make functional nanomaterials, aspects that you can turn into an application, a product. So you can think about these kind of things: micelles, membranes, metal organic frameworks, active surfaces. The luminescent nanoparticles, they're also used a lot in biology. So you can attach a nanoparticle to a protein, and thereby you can see the location of that protein in the cell. So you can monitor processes in the cell in that way. These are the parts of the book to which this is related. Questions? Are there
Yes,
questions?
I have a question.
Yeah.
For the quantum dots. So because of the size of the box, if you have a smaller box, you get more blue shifted, but isn't it also because a larger nanoparticle has more atoms, so it has more energy levels, which are therefore closely packed? So isn't that also a reason why larger nanoparticles have a smaller separation between energy levels?
You can coat the nanoparticle with an extra layer, so you can make them physically bigger, but it is the size of the semiconductor that influences the emission color. Of course, it's not possible to make a bigger particle without changing the number of atoms. They are called quantum dots because of this quantization effect, that the size determines the properties, in this case, the emission properties. The number of atoms has to change, but it is not only, if you make, for instance, gold, then you can change the size of the gold nanoparticle, but then only the 520 nanometer transition, that will grow in intensity. If you use gold, then there is not this same size effect. So it is not that only the number of atoms makes this effect.
Wait, but for gold there is a size effect, right? So if you have a higher concentration, you have a, well, because I learned about quantum dots before, and then there was a size effect for gold particles.
The intensity of the 520 nanometer absorption band, if you go from 1 to, let's say, 20 nanometer, that's only the intensity changes. But if you make them even bigger, then they can shift to the blue, to the red. They can become more blue colored.
Yeah. Okay.
So it is not only the number of atoms.
Yeah, okay.
A collective movement of oscillating electrons on the size of the semiconductor quantum dot.